Okay, class, it's an exciting day here today because we get to start a new chapter. Chapter 10, we're taking a look at genetics. We're taking a look at the patterns of inheritance here. And the person that's considered the father of genetics is Gregor Mendel. He was an Austrian monk in the 1860s. He experimented using garden pea plants. There was a lot of advantages to using these because you can have thousands of them in a little small area. They have distinct traits that he looked at: tall or short, round and wrinkled on the seed shape, seed color, yellow, green, et cetera, et cetera. They are very cheap. The turnover time for reproduction from one generation to the next is fairly quick. So all these were advantages to use a pea plant. If you remember your punnet squares, if I take a look at, first of all, what's the pea generation? So let's say I'm taking a look at the trait tall or short. And first of all, the capital T is dominant. It's the dominant allele. Allele is a different expression of a trait, so we're taking a look at height here. Tall is one expression or one allele, and short is the other one. The recessive allele is short. The dominant one covers up the short one if they're together. So if I take a dominant, let's say that came from the male, and then the recessive from a female. Remember, I take this trait here and drop it down in both boxes, and then the same with this one. And then I take this one and take it across to both of them and do the same with this one. So again, I start out with the P generation. That would be my this parent and this parent. What I get is the F1, allele. It stands for generation. So this is the first generation. And if I take a look at those, I get, if I look at the genotype, what's genotype? It's the genes. I have 100% that are capital T, small t, or 4 out of 4. If I take a look at my phenotype, in other words, what they look like, and if I go tall to short, I have 4 to 0, or 100% tall. So now if I take that F1, remember the ones in the boxes are the F1 generation. If I take them and cross them, so in this case I'd have a capital T, small t for one parent, and the same for the other parent. If I drop that capital T down, and this small t down to both boxes, that capital T across to both, and the small t across to both. Now all of a sudden, if I take a look at the genotype, I have some different genotypes. I have capital T, I have capital T, small t, this one and this one, and I have small t, small t. And if I count those, I have 1 to 2 to 1. Or if I put it in percentages, I have 25% of them would be tall, homozygous tall, and 50% of them would be heterozygous for tall, 25% of them would be homozygous recessive or short. And if I take a look at my phenotype, tall to short again, I have 1, 2, 3 tall and only 1 short. So 75% are tall, 25% are going to be short. So what does a Punnett square show you? It shows you who the parents are, the offspring are, and it shows you all the possible combinations, and it gives me the probability of those combinations. Nothing else, when I cross in this first one, if I cross those two, nothing else can happen but that. And I crossed it in the second two, nothing else can happen but those. and it shows you the percentage of each one of them. Okay, again, these are punnet squares, and it's a monohybrid cross because we're only looking at one trait. The law of segregation, each individual has two factors. Why does it have two factors? Because it got one from its mother and one from its father. So each one has two, just like the top. Right here, there's two characteristics there. This parent had two, and then this offspring had two, that one had two, so on and so forth. Every one of them had two traits for tall, two different alleles for being tall. The factors separate during the formation of gametes. So when we did meiosis the other day, and when we said that you have chromosome number 1, let's say that from on this particular one, the capital T, so if I take a look at this one right here, this one right here, I have a capital T on one chromosome and a small t on the other one. Well, when they divide in meiosis in the final step, I'm only going to have one trait over here, and in this case, the other trait over here in those eggs or the sperm, whichever one. They segregate during the formation of the gametes. Also, each gamete contains only one factor. Like I said, this one had a capital T, this one had the small t. Fertilization gives each new individual two factors. So we had this is in making an egg or a sperm. There's only one trait, but when it would form a fertilized egg, it would combine with the other set, and once again, you have a set of chromosomes. So we already took a look at what a genotype was. That's taking a look at, these. That's the genotype. What's homozygous are the same. So, up here in my example, which one of these are homozygous? This parent was homozygous dominant. This parent was homozygous recessive. This offspring was homozygous recessive. That one was homozygous dominant. So what's heterozygous? It's one trait of each. That's that one, this one, this one, this one. Any of them that have one of each. What's a heterozygote look like? Right here. Here's my heterozygote. As you can see, they're just allele uneven. Okay, sorry. For the assignment tomorrow, you need to write heterozygotes at the top when you hand it in. That will be worth 2 points. So if you don't do that, I will take 2 points off. So I'm making sure that you're watching this video, okay? And we talked about those already, but the phenotype is what they look like. In this case, it would be the looks of these wild made-up sheep. Or in our case up here, it'd be tall and short. That's the phenotype. For your assignment tomorrow, you'll be responsible for doing some punnet squares like we did here and giving the genotype and the phenotype of them. Okay? Don't forget to write the heterozygotes on the top. All right. We'll see you tomorrow. Thanks. Bye.